Bill Anderson here? That might be important.
Yeah, he is.
Yes, sir, right here. Hi.
Then we are good to go.
All righty. Well, welcome everybody, and thank you all for coming for Peter's presentation of his dissertation. The dissertation is titled Spatial and Temporal Trophic Dynamics and Energy Fluxes in Response to Ecosystem Engineering and Invasive Species. Although Peter and I have had a running conversation about exactly what the title is going to be, so we'll see what he actually uses. So Peter earned a BS degree in marine science with a minor in mathematics at the University of Tampa in 2015. From there, he enrolled at the University of Southern Mississippi, where he studied for a year before transferring to enroll in our PhD program at FIU in the fall of 2017. So while in grad school, Peter has been very productive. There we go. He's published 2 peer-reviewed papers, and he has a 3rd in review at the journal Food Webs. While in grad school, he's also taken lots of opportunities to present his research at national and regional meetings. And in fact, he's given 7 different seminars. Uh-oh. What do you think?
Can we meet?
Please mute your microphone. Okay. All right. His research he's going to talk about today was primarily funded by the Critical Ecosystems Study Initiative, or CZ. Some people may have heard it called that, from the Everglades National Park. But he's also received an Everglades Foundation FIU for Everglades Scholarship in 2019. And he has received an award from the FIU Institute of Environment, so one of their student grants in 2020. And in fact, finally, in February of 2023, Peter's going to be starting as a postdoc in Karen Aloff's lab in the School for Environment and Sustainability at the University of Michigan. So Peter became interested in the ecology of invasive species as an undergraduate, and he came to FIU to pursue that interest through studies in the Everglades. So while conducting his work on invasive species, he's focused on the impact of invasive species on aquatic food webs and community structure and function. So today he'll be telling about the product of his past 5 years of research, and I know we're all excited to hear about it. So Peter, please go right ahead.
Yeah, thanks, Joel. That was great. All right, let me share my screen here. And we can get things rolling. So here's yet another title. It's different than the one Joel said, and I don't think either of them are going to be the final title, but we'll just jump right in anyway. So this is primarily a dissertation.
Peter, the screen is not showing. I'm just seeing all the people on.
Oh no. Okay. One click away.
There you go. That's got it.
All right. Get this back in the presentation mode. All right, you've seen the PowerPoint? All right.
Yeah, that's it.
So this is a food web. Why study food webs? Why are food webs important? Why do we care? So each node in a food web represents a population. At the bottom, we have producers, and you move up the trophic levels to these higher order consumers. The nodes are populations. Sometimes they're communities. These ones in boxes in yellow are really communities of zooplankton, communities of fishes. And then, of course, the entire food web as a whole is also a community. And then in 1942, Lindemann gave us the trophic dynamic aspect of ecology, where he showed us how food webs affect biogeochemical processes, primary production, and secondary production. And work that's a little more recent than 1942 has connected food webs to ecosystem resilience. This figure at the top, this top circle, shows that as you move up the y-axis, increasing trophic position organisms rely on carbon sources from a variety of sources. The diversity of that food web structure that gets them their energy, that's represented in this bottom left circle. And then that diversity increases ecosystem stability, which is represented in the bottom right circle. So why study food webs? Because they unite the fields of population and community ecology with ecosystem ecology, and they provide insight into these ecosystem level patterns. And just like these folks unite to form Captain Planet, different aspects of ecology are united in food webs. So today we're going to look at three drivers or three controls and dynamics of food webs. The first is spatial and temporal variation. How do food webs change in different habitats in different seasons? And ecosystem engineering, specifically, how does the American alligator influence trophic dynamics? And then what about invasive species? As Joel mentioned, that's sort of been the focus of this, the motivation behind this. And this is the African jewelfish. So what does all this look like and how do we put it together? We have habitat variance in space, temporal variance in across seasons. As you put these together, you get fluctuation in abiotic conditions across time and space. Oh boy. Sorry, folks. All right. So fluctuation in abiotic conditions in time and space. That fluctuation in things like light and nutrients to temporal and spatial fluctuation in what's biotically available to consumers. And this in turn leads to shifts in diet. Additionally, ontogeny, as these animals grow, their diet changes, just like us. And then these shifts in diet can lead to two different responses that will be in this blue box that I'll walk through real quick. Trophic displacement, that simply means a shift in diet. Trophic dispersion is a change in the trophic niche. Is it getting bigger? Is it getting smaller? Shifts in trophic position, has the trophic position increased? Has it decreased? And then also shifts in basal resource use. So this gradient from autotrophic green photosynthetic energy to brown heterotrophic microbial energy. And then when you put all of these together across the community, across multiple species, you're talking about a whole food web and the structure and function of that food web. And then as I was just talking about earlier in the introduction, that in turn feeds back and influences ecosystem stability and resilience. So in chapter 1, we're going to ask, how do food webs change in space and time using this framework? In chapter 2, we'll throw in ecosystem engineers that influence the physical habitat, specifically the American alligator, and ask how does that change? Could everyone please mute themselves on Zoom? Let's see. Just make sure everybody's muted if you're on Zoom, please. And then in chapter 3, we're going to add in invasive species, specifically the African jewelfish, and we're going to ask how have African jewelfish changed trophic dynamics and energy fluxes in the Everglades. So with that, step through the wormhole with me into chapter 1, and we'll be asking how do food webs change in space and time. So we're back to our original food web diagram here. We're all very familiar with these diagrams in ecology. You see them all the time, all over the place. What we might not realize is that we make an assumption every time we put one of these diagrams on the screen or talk about one of them, and that is that this is static in space and in time, and that these relationships don't change. So in this first part of the presentation, I want to ask all of you, what do we miss by assuming food webs are static in space and in time? From a temporal perspective, seasonality is probably the most ubiquitous driver of temporal variation. And this was recognized by Charles Elton in his 1927 book, Animal Ecology, almost a century ago. Similarly, habitat heterogeneity, which as you see in streams on the top and river floodplains on the bottom, leads to this spatial variation in which reduces the strength of individual interactions between individuals and species, and also creates a resource asynchrony across the landscape. And both of these things contribute to increased ecosystem stability and resilience. Despite all of this, most interaction networks in ecology, including but not limited to food webs, are treated as static in space and in time. And there's some great reasons for this. This photo is covered in ice, and apparently so is a lot of the world in the winter. We all came to Miami, well, most of us came to Miami, at least in part to avoid the snow and the ice. It's a cold day in the Everglades when it's 40 degrees and you're on the airboat. Can't imagine trying to work in a pond in the winter. And then, of course, resources are limiting and we have to integrate data across larger spatial and temporal scales than maybe is ideal. But what do we miss when we do this? And in this first chapter, we looked at a whole bunch of different response variables, but today we're just going to focus in on trophic position and trophic niche and how they changed in space and in time. So most of this work that has been done looking at temporal variance in trophic position and trophic niche has been done in this river floodplain ecosystem where You see on the left, that's the normal condition. In the wet season, the river floods, oftentimes into a flooded forest. Think Amazon, think rainforest. So if you're a fish, if you're an aquatic consumer, what resources are pulsing for you depends on what you eat and your functional feeding mode. So it's no surprise that when you look at the literature, you get a mix of directions of effects for different fishes, and we'll run through a few examples. This first fish in the wet season, the river floods and it gets access to these terrestrial arboreal insects. And in the dry season, it's eating green algae. So this leads to a higher wet season trophic position. Next fish does the opposite. In the wet season, the river floods and it goes and it eats seeds and fruits out of the trees. Whereas in the dry season, it's eating these aquatic macroinvertebrates. And then this final fish is an omnivore. And then it's still an omnivore. It doesn't really have a change in diet depending on the flood condition. So our system, Everglades, is a great place to study these dynamics for two reasons. The first one is space. The Everglades is often defined or described as a landscape mosaic, and that adjective just highlights the spatial variance. You can see some of the different habitats pictured on the slide. And the second is time. We have distinct wet and dry seasons. You see the dry season on the left, the wet season on the right, and how the change in available habitat. So what do we miss when we assume food webs are static in space and time? How do trophic positions change? How do trophic niches change in size? And how do they change in their overlap with one another? So how did we do this? First, this is Bill Loftus. He collected all of these data in 1977 to 1981. So this chapter would not have been possible. And the third chapter, this is the pre-invasion baseline for. So a lot of this work wouldn't have been possible without Bill. And I should also note that 1981 is the year my parents graduated from high school. So Bill collected data in both the dry season and the wet season, and he collected data in three different habitats. He collected data in these blue spikerush dominated sloughs and the green sawgrass dominated ridges, as well as alligator ponds. So alligator ponds themselves are quite variable. You can see two different ones on the top, and this third pond on the bottom, you can see this full gradient from the pond, the spikerush marsh, the sawgrass ridge behind those two guys messing around out there. So this resulted in a total of 32 species of fishes being collected. They were divided into size classes based on their length. That resulted in 53 size classes for a total of about 3,500 stomachs that were analyzed. The target sample size per size class in each habitat in each season was 25, and we ended up with an average of 16. So to calculate trophic position, we assigned these more specific prey groups in this table into these broad functional groups, and then each functional group was assigned a trophic score. We plugged these into this equation from Adams et al. In 1983, which basically just says that a given individual is one trophic position higher than the weighted average of its prey. And then we compared these across habitats and across seasons using permuted analysis of variance. Now for trophic niches, we took a little bit of a novel statistical approach to modeling trophic niches from stomach content. First, we took all those different prey categories that are each a column in a matrix, and we shrunk that down to two columns using non-metric multidimensional scaling. And then we put those two columns into the cyber package in R, which generates these ellipses using this Bayesian methodology. And if you're at all familiar with the cyber package, you might realize that SI stands for stable isotopes, and we're talking about stomach contents. But what stable isotopes really are ordinations of diet that occur in nature. So here we're just doing an ordination of diet that is mathematical. So it's very analogous and functionally similar. And we're talking about niches in NMDS space and ordination space instead of this isotope space. And then we can directly compare these, their size and their overlap among habitat season levels using Bayesian inference and the tools in the cyber package. What do these changes in trophic niche look like? Here we've got the eastern mosquito fish, Gambusio holbrooki. So this will be our example fish. We did our ordination. This is where these consumers fell out in ordination space. These are their prey items, and that's where their prey items fell out in ordination space. And then this blue circle is the resulting ellipse that came out of our cyber modeling. Now we'll say that this is the wet season in the pond. And now this gold ellipse is the dry season in the pond. And you can see that ellipse is a little bigger And that's because in the dry season, we've added 2 additional, diet items, increasing the size of the trophic niche.
You shouldn't have to take, I had 2 people coming back.
Oh, I didn't know you did.
Well, Olivia will be coming back.
Could everybody please mute on Zoom? Could everyone please mute on Zoom?
She's really young. She's 37 or 38.
Just getting ready to start now, but.
You,
Sure.
Peter, you might be able to mute them yourself from your computer.
Yeah, there's a lot of folks on here.
Teresa, it keeps. I'm gonna
Hey,
get out of
Peter,
it.
I've got it and I'm trying to mute him.
All right, thank you, Evelyn. All right, lots of excitement. So mathematically, statistically, we calculated whether these ellipses were smaller or larger than each other by comparing the posterior distributions of these 2 ellipses, the areas of these 2 ellipses. And if the probability that one ellipse was greater than the other exceeded 95%, we called that an increase in niche size, and the opposite if it was smaller. You can also have changes in niche overlap. So in this example, between these 2 ellipses, there's only one shared prey item, that red chironomid larvae that's at the bottom of the left and at the top on the right. So they each have 3 diet items. Their niche is the same size, but they have divergent diets. So those ellipses have moved apart in the ordination space. So we have a shift in overlap, but not a shift in size. And shifts in overlap were calculated if the intersectional area, where the Venn diagram there overlaps, was less than 5% of the total union area of the 2 ellipses. And finally, of course, you can have a shift in size and a shift in overlap. So here again, there's only one shared prey item, but many additional prey items in the gold ellipse. So an increase in size and a shift in overlap. What did these results actually look like? And we'll start with trophic position. How did it change in space and time? So again, we had 53 species size classes. About a quarter of those had a shift in trophic position across space and or time. 6 of these were habitat shifts. So in the same season, the wet season, between the pond and the spike rush marsh, there was a shift in trophic position. 2 of these were seasonal shifts. So if you're in the pond, it's the same habitat, but you had a shift in trophic position between the wet season and the dry season. And the remaining ones were an interaction of habitat and season. So why did these trophic positions change? We identified these 5 feeding modes as our potential drivers. Piscivory is a subset of carnivory here. And then diet plasticity, what we mean by this is if you think back to that table about trophic position, and we lumped all those prey items into their broad functional groups, you can imagine a situation where it's possible to have a change in trophic position, but your diet proportionally is the same proportions of those functional groups because you're feeding on different members of the functional group. So that's what we mean by diet plasticity. And we found that carnivory, piscivory, and diet plasticity were the 3 most important drivers of shifts in trophic position. So let's move on to shifts in trophic niche size and trophic niche overlap. And again, we had 53 species size classes. So there's a few ellipses that were generated. These are some season comparisons, some more season comparisons, and some habitat comparisons. So we're going to dig into these panel by panel, ellipse by ellipse, and talk about the species specific drivers of each one of these changes. Or we can just talk about the eastern mosquito fish, since I don't want to keep everybody here for the next 4 hours. So we'll just run through what these changes look like relative to those examples that I gave everyone. This top middle shift is a change in size, but not a change in overlap. So the blue ellipse is smaller and it's moved a little bit to the right, but not enough for a statistical shift in overlap. Bottom left here is a change in overlap. The ellipses have shifted apart, but they're roughly the same size still. And these 3 panels are both a change in size and a change in overlap. And the bottom right panel that I haven't referenced at all is neither a change in size nor a change in overlap. And of course, we have these all again for our habitat comparisons. And since there are so many size classes, I'm just going to run through some summary statistics across all the comparisons in space and time. So what we found were 66% of our comparisons demonstrated spatiotemporal shifts in their trophic niche size. 86% of comparisons had overlap less than 5%. And again, the drivers of these, we looked at the same functional feeding modes, and we found carnivory, piscivory, and diet plasticity were once again the most important drivers. And this time there was an increase in herbivory. If you recall, before it was only 7%. So in conclusion, for chapter 1, we found relatively few shifts in trophic position. Shifts in trophic niche were common. Shifts in trophic niche overlap were ubiquitous. And the primary drivers of these trophic dynamics were carnivory, including piscivory, diaplasticity, and detritivory. But what would we have missed if we had assumed that this food web was static in space and time? We would have missed a quarter of our species size classes having a shift in trophic position, most species size classes having a change in niche size, and the vast majority having a shift in niche overlap. So far, we've talked about how habitats and seasons influence food webs. And now we're going to add in ecosystem engineers, American alligator, and see how they influence trophic dynamics. So what is an ecosystem engineer? We can all probably think of a bunch of examples. There's kelp and kelp forests. There's these like rocky, spiky, stony things. There's a coral. I'm never really sure. I think I've seen people talk about it here before. Seagrass, beavers making dams, these floating environments, elephants, and our very own red-bellied woodpecker right out here from driving into BBC that maybe you saw before Hurricane Ian blew its tree down. But these are all ecosystem engineers, and they can influence the food web in a couple of ways. First, on the top, they can influence the nodes in the food web by creating this environment, and then that can influence the population fitness, other things like that influence the nodes, and then also the links. So you can imagine that if you're engineering a habitat that's open, doesn't have a lot of structure, maybe now it's easier for the predators to catch prey, where also the opposite could be true. If you engineer a habitat with lots of structure, you could inhibit predation. And then ecosystem engineers can also impact food webs at a subset of different trophic levels, one trophic level, a food web compartment, or the entire food web, especially in situations where the engineer creates the entire habitat. And that's exactly what happens in this example with the sociable weaver. They create these massive colonies that you can see in an arid landscape that, like South Florida, has a wet season and a dry season. So there's a spatial gradient of environmental stress and a temporal gradient of environmental stress. And these colonies are a respite for both of those things and biodiversity hotspots year-round. And this idea where ecosystem engineering ameliorates environmental stress is known as the stress gradient hypothesis, which states that the relative frequency of facilitation and competition will vary inversely across gradients of physical stress or ecosystem productivity. And we have the opportunity to test this hypothesis right here in South Florida with the American alligator. They dig and maintain these alligator ponds, like this one picture on the right. And on the left, we have a picture that I took in February of 2017, which may be the beginning of a brand new alligator pond by this fellow here. Now, ponds themselves can be quite variable. The pictures of alligator ponds I've shown throughout were ponds we visited in this study. There's 6 of them pictured here. Some previous work by our group led by Bradley Strickland documented consumer and producer populations, as well as flock and periphyton and consumer nutrient levels across this gradient. As you can see visually, there's a distinct producer community in each of these habitats. From the pond on the left, what we call the near pond, this dense halo of vegetation surrounding the pond, that's also a result of alligator engineering. And then the adjacent spike rush marsh. And most interestingly, perhaps, is we found a phosphorus gradient of increasing phosphorus as you move from the marsh to the near pond to the pond, with the pond having the most phosphorus of these habitats. So when we go to ask about the stress gradient hypothesis, most of these studies look at biodiversity. They see an increase in these engineered habitats, and they say, well, competition must go down to facilitate that increase in biodiversity. It's a little bit of the ghost of competition present, if you will. So we didn't measure competition, but we're going to try to get an assist from the niche variation hypothesis, which states that competition and niche size are inversely related. And we'll just walk through this diagram of our predictions of how these two hypotheses coincide. So on the x-axis, we have environmental stress, and on the y-axis, we have interaction intensity. So first, the stress gradient hypothesis tells us that as environmental stress increases, we also see this increase in the intensity of, or the strength of the facilitative interactions, and the opposite for competition. Now, when we shift to the niche variation hypothesis, as competition decreases, niche size, these blue circles, increases. So when we have lower competition, we have a larger niche area, and vice versa. So for this chapter, we have a few predictions. that consumers will undergo dietary shifts in these engineered habitats. Trophic positions will increase in deeper habitats, specifically in the ponds. And then these elevated nutrient levels from this engineered nutrient gradient will generate bottom-up effects that lead to increased autotrophic energy use. And then finally, that trophic niches will increase as environmental stress increases in engineered habitats. Now, again, we're just going to focus in on this final prediction, and that increasing stress is as we move from the wet season to the dry season. And what we expect is that niche areas will increase in the dry season relative to the wet season in ponds, but decrease in these other two habitats. So to do this, we visited 10 alligator ponds, 5 in each of the major drainages of the Everglades. Shark River Slough is that one to the northwest, and Taylor Slough down to the southeast. And at these sites, we collected everything we could across the whole food web. We ended up with stomach contents from almost 1,700 individuals and almost 4,000 stable isotope samples across 106 different consumer groups, most of which were identified to species. And then again, these were divided into species size classes based on length to account for ontogenetic shifts in diet. We modeled these niches the same way in chapter 1 with the cyber package, and then again, compare them across time and space using the same methods. So what did we find? We've got our predictions up here on the top right. We'll start with the results based on stable isotopes. So far, so good. Niches increase in the dry season in the pond. They decrease in these other two habitats. And then I looked at the results from the stable isotope data, and they did the exact opposite of what we predicted across the board in every habitat. So that was great. So what's going on here? The stomach contents supported the stress gradient hypothesis, while the isotopes were the opposite of our predictions. So for this, perhaps we can get some idea of what might be happening from this meta-analysis from Alofs and Jackson, where they looked at consumptive versus competitive resistance to invasions, the aspects of the biotic resistance hypothesis. So they looked at invasive producers in freshwater ecosystems, invasive consumers, and the same in marine ecosystems, and then invasive producers in terrestrial ecosystems. And on the x-axis, you have the effect size of biotic resistance from these different effects, consumption and competition. And yes, this is about an invasive species and invasion biology, but invasion biology looks at case studies of community ecology where you add a new species to the community. So these same effects governing invasion resistance also influence broadly community structure and how communities are structured. And what you'll notice is that in these aquatic ecosystems, the consumptive effects are larger than the competitive effects across the board. And if you focus in on that top part in the freshwater ecosystems, you'll notice that those 95% confidence intervals don't even overlap. So in freshwater ecosystems, it seems like consumptive effects are much more important than competitive effects. So let's circle back. How does this link in to what we're talking about here with these niche areas? Well, stomach contents reflect a relatively short time period, about 24 hours, maybe 48. So perhaps over those shorter time periods, we're seeing what we expect because at a smaller time scale, competition is governing that feeding behavior. But when you move to a larger time period, like those reflected by stable isotopes, which is typically 3 to 4 weeks, it could be that consumptive effects, specifically predation and avoidance of predation, is leading these consumers to forage across different habitats, which would lead to this increase in isotopic niche that we saw. Reviews in the literature have also shown that there's this trade-off where competition has a greater effect on growth and predation has a greater effect on survival. So perhaps this differential habitat use is to optimize this trade-off, particularly in the dry season, as fishes, especially larger predators, concentrate in these ponds and are ramping up the predation pressure. So overall in this chapter, we found mixed support for the stress gradient hypothesis via the niche variation hypothesis. We supported it with stomach contents. We found the opposite with stable isotopes, but it seems like perhaps what we really found was an interesting dynamic about these consumptive and competitive effects. And overall, Mielikson and Torberger, in their review in 2010, point out that the important piece of the stress gradient hypothesis is not the additive effects of competition versus facilitation, but whether or not the species has an increase in its fitness. A lot of these fishes, especially the larger ones, use these ponds as dry season refuges. And when the alternate option is no habitat, having habitat increases your fitness. So we've added ecosystem engineers, and there's one more to go, invasive species. How do these add into the mix, and how have jewelfish changed Everglades webs and trophic dynamics? We'll start with an example from a different system. This is Yellowstone National Park, and on the left will be the uninvaded food web. On the right of this vertical gray line is the invaded food web. This is the native trout here, and you can see that it's consuming some invertebrates here. The size of these arrows is proportional to their dietary contribution up the food web. These trout are in turn consumed by some mammals and some birds and some more birds. This is the aquatic food web in the invaded lakes. This new trout, the lake trout, has come in, is both taking prey from the native trout and consuming the native trout. And now when we're on this side of the figure, some of these animals will be smaller. Like that native trout is smaller, and that size difference reflects the relative abundance pre and post invasion. The lake trout is also now it's also consuming this other catastomid in the bottom right, which is part of the otter diet. So now that is less energy going from those fishes to otters, less native trout going into bears, causes bears to feed more on elk. Similarly, we get less trout to these other avian consumers, and then the raptors are in turn feeding on other birds. If you look closely, you'll notice that this gull is not here in the post because it no longer nests in Yellowstone. And you'll notice the osprey is smaller because its relative abundance has decreased. This is what one freshwater fish can do. So yes, freshwater fish, non-native freshwater fish can be bad. These effects have recently been formally called the Trophic Disruption Hypothesis by Wainwright and colleagues in a paper that came out last year. And they say that invasive species disrupt food webs in two ways: trophic displacement, which again is a shift in diet, and trophic dispersion, which is this change in trophic niche size. And you can see that in this picture, this diagram from their paper, where they looked at lakes that were uninvaded, the reference, been invaded for a shorter period of time in the mid, and then lakes that had been invaded for decades in the late. And they have these groups by the blue and the red are the two trout species, and the other colors, they've functionally grouped these fish species. And you can see that those ellipses change in size and also their location in isotope space as you go through time. So that's trophic dispersion. Now, this hypothesis doesn't have any specific predictions about the direction of these changes, but we can turn to the literature. Our friend from chapter 2, the niche variation hypothesis, which again says that interspecific competition, like that from invasive species, will decrease niche size. And then some work by McMeans et al. Shows that consumers become more generalist in response to avoid invasive predators. So now I'm talking about changes in food webs that sound a lot like the changes I showed you in chapter 1, where I tried to convince you that that variation improved ecosystem stability and resilience. And now I'm talking about similar dynamics in a negative context. So it begs the question, what's different about these two different sources of variation in food webs? Perhaps they have different frequencies or different magnitudes. So we're going to look into that as well. So what about the Everglades? How does this all have anything to do with the Everglades? Well, this is a very data-rich ecosystem. The map on the right shows all of the sites that have been monitored back to 1996 through the Modified Water Delivery Program. And folks have been going out 5 times a year since 1996 with these throw traps and throwing them at these sites. At each of these dots, there's 3 plots. And at each of those plots, there are 7 throws per plot. And like I said, they've been going out 5 times a year for quite some time now. 2 of these sites go back even further to 1978, which, if you recall from chapter 1, coincides with the stomach contents collected from 77 to 81. So we look at the community composition through time from those 2 sites that go all the way back to 1978. We have, we'll talk more in a couple of minutes about why we picked these groups, but these groups have this variation sort of along this diagonal here from bottom left to top right. And then all of a sudden in 2012, we got variation in this other direction, and the ellipses since 2012 do not overlap with any of the ellipses from the long-term data prior to that. So what happened in 2012? This is a hydrologically pulsed system with distinct wet and dry seasons, and it's also been the focus of the largest restoration program in the world. So perhaps it's got something to do with hydrology and changes in hydro period. So those same time periods as the ellipses, we looked at the hydro period, which is the mean and excuse me, the number of days with water greater than or equal to 5 centimeters each year. We compared those across these time periods, and it was not significantly different. So there's just those time periods again in our study in 2018, 2019. So perhaps it's the result of biotic interactions and the invasion of all these fish species. Roughly 100 fishes have been introduced into the fresh waters of South Florida over the past 50 plus years. 18 of these have made their way into Everglades National Park, and 15 of them have become established. So that's 15 non-native fishes relative to our 35 native fishes. So what happens with the relative abundance of these fishes through time? So this is using the data set that only goes back to 1996, but has a wider spatial coverage. And these are the sites from Shark River Slough. And what you'll see as we go across this diagram is that there are 3 non-native fish that are really major players so far. Everything else is in yellow. And if you didn't bring your binoculars, you probably can't see any yellow on there. We have the Mayan cichlid in purple, the African jewelfish in blue, and the Asian swampy in this teal. And what you'll see on all these sites is this spike in African jewelfish between these 2 red lines. And that first red line is 2012, when we saw that shift in community composition. This first black line is the year 2000, when the jewelfish invaded the Everglades. Their population decreased precipitously here, and this is the end of 2017 is this red line. The vertical blue line is when we sampled in the wet season, and the brown line is when we sampled in the dry season. So a huge spike in the relative abundance of these jewelfish. Most other species on the top left there are at very low densities and not likely to cause effects. And then at the time of the changes in community composition that we saw, we did not see spikes in Mayan cichlids, and the Asian swamp eel was just making its way into this part of the Everglades. When we performed this study, we actually caught the first one in this region of the Everglades during our study. So what about biomass of these fish? On the y-axis here, we have the relative biomass of African jewelfish. And what you'll see is at site 6, during the peak of that boom, they were more than 50% of the biomass at that site. And at these other sites, you can see it in excess of 20%, in excess of 40%. So high relative biomass at these sites. We have some data from another long-term monitoring program that samples at a much larger spatial scale each wet season. We're going all the way almost up to Lake Okeechobee here. It's probably right about here. And we have this Loxahatchee Wildlife Refuge coming down through the water conservation areas into Everglades National Park. Each of these dots is a site that was sampled, and the circles on them is the relative abundance of non-native fishes. They're color-coded by the dominant species, and green is the African jewelfish. The companion plot next to it now is the total count of fishes at each of those same sites during the same sampling event. And what you can see is a pretty stark inverse relationship between the relative abundance of these African jewelfish on the left and the total counts of native fishes on the right. So it's not just isolated at small spatial scales. And what we see as well in this figure, we have different species along the x-axis, biomass on the y-axis. Their pre-invasion biomass is in red in 1996, well after the invasion during the peak of the jewelfish boom in 2016, we have their biomass in blue. What you see are precipitous declines in many native species in their biomass, some much lower. You look at the eastern mosquito fish here, some of these are gone, and then of course this increase in biomass of the African jewelfish. So we come back to this first figure that I showed you about the community composition through time. So now we know that in 2012, what starts to separate these ellipses from the historic fluctuation in community composition is the African jewelfish. And what you see is this is the African jewelfish right here. So this variation shifted from differences in the relative abundances of species sort of in this axis to differences in this axis, a range from jewelfish to eastern mosquito fish. So what are we trying to do here? Our goals are to evaluate that trophic disruption hypothesis that I talked about, specifically its premises of trophic displacement and trophic dispersion. And we predict that native species will eat less of the prey items that jewelfish are also predating upon, and that trophic dispersion will take the form of increased niche sizes as we have more species trying to access the same limited resource pool. We'll compare the frequency and magnitude of spatiotemporal variation, like we saw in chapter 1, to trophic disruption. What's really different about these two phenomena? And we just sort of have a null prediction here that they'll have a similar frequency and a similar magnitude. And then we want to know if the trophic disruption hypothesis can be extended to include other response variables like trophic position, shifts in basal resources, and energy flow. And we expect that native taxa will rely primarily on detrital energy, like has been shown in previous work. And we expect that African jewelfish will have shifted energy fluxes through the food web. So more stomach contents here. So this chapter uses the data that overlaps from chapters 1 and chapter 2. So here, post-invasion, we have just shy of 1,000 stomachs from 46 different taxa. Again, the pre-invasion data are 1977 to 1981. Post-invasion, wet season 2018, dry season 2019. These are pond and spike rush marsh habitats, the two shared habitats, and we have these in both the wet season and the dry season. And they're divided by species size classes, and we have spatiotemporal variation in the stomach content data set. Stable isotopes, we got 2,200 post-invasion from 90 taxa. The pre-invasion stable isotopes were collected in 1994 from only wet season marshes. So we're using our complementary post-invasion isotope data from wet season marshes in 2018. These are to species level because the pre-invasion data was also to species level, so we're matching that. And then one habitat, one season, no spatiotemporal variation from the isotope data. So first, trophic displacement. We looked at the diet communities of species size classes, and we compared those with pairwise permuted analysis, permuted multivariate analysis of variance between pre and post-invasion for a size class within a habitat, within a season, and compared those pre and post-invasion. And then similarly, pre-invasion, and then again, post-invasion, we compared how a size class changed across time and across space. And then we compared the frequency and magnitude of those effects pre and post-invasion. With dispersion, niche areas, we did the same thing we did in chapter 1 and 2, generating these ellipse areas, comparing them across space and time. And again, we did the same thing with the stable isotope data as well. Trophic position, we used our same equation for the stomach contents, and those statistical analyses also matched chapter 1, permuted analysis of variance and pairwise post hoc tests that were permuted. Stable isotopes, we modeled those trophic positions using this R package, trophic position, nice name. And within that, you can do these direct pairwise comparisons of the trophic positions. And this modeling approach also produces this metric called alpha, which is the proportion of the nitrogen isotope that comes from your basal resources, your two end members. So in this case, alpha values greater than 0.5 indicate more brown heterotrophic energy, values less than 0.5 or more green autotrophic energy. And then finally, we modeled energy flux through this whole food web. And we did that using the flux web package in R that incorporates biomass, metabolic losses, assimilation efficiency, and diet relationships. We got the diet relationships from the stomach contents from this work, community composition, and the long-term monitoring data. We converted these into biomass in the form of ash-free dry mass. Metabolic losses and assimilation efficiency were derived from the literature. And then pre versus post-invasion marshes, we were able to do this energy flux modeling for both of those in the wet season and the dry season. So our first objective, did we see trophic disruption and our native species taking fewer prey that the jewelfish would take? We found that 42% of species size classes underwent trophic displacement. And this was largely driven by a shift in the relative consumption of omnivorous invertebrates, carnivorous invertebrates, detritus, and herbivorous invertebrates. So our second prediction, trophic dispersion would take the form of increased niches. What did we find? From stomach contents, we found that 54% of species size classes demonstrated trophic dispersion post-invasion. 73% of these were increases in niche size with a mean absolute value of percent change of 278%. And you can see in this figure here, we have percent change along the x-axis and the frequency on the y-axis. You can see that some of these percent change values are quite large, sometimes exceeding 2,000%. And the functional groups most impacted were invertivores and omnivores, which are the two functional groups predominantly competing with and or being predated on by jewelfish. When we shift to trophic dispersion from stable isotopes, we found that 46% of species size classes underwent trophic dispersion post-invasion. 75% of these changes were decreases in niche size, with a mean absolute value of percent change of 357%. So again, quite large effect sizes. And with that, we'll move on to our second point. What was the frequency and magnitude of the spatiotemporal variation relative to trophic disruption? We found that they actually had identical frequencies in this case. Magnitude of spatiotemporal variation on average was 131%, while the magnitude trophic disruption was 278%. So that's more than 2 times the magnitude for trophic disruption relative to the spatiotemporal variation. So now we'll test whether or not the trophic disruption hypothesis should include these other trophic response variables. And we thought that native taxa would rely primarily on detrital energy. So for shifts in trophic position, just like in chapter 1, there were relatively few of these compared to shifts in diet and shifts in niche. However, we found that 43% of species size classes had a shift in their alpha metric. So you can see those here. Again, this is a gradient of brown energy, higher on the y-axis, greener energy, lower values on the y-axis. We have pre-invasion in purple and post-invasion in green, and we have different consumers down here along the x-axis. And what you can see is that every consumer, whether it was a statistical shift or not, decreased in alpha, which is to say that it increased in its autotrophic, its photosynthetic energy that it was consuming through the food web. And then finally, did African jewelfish change energy fluxes in our ecosystem? So here we've got several very busy plots of food webs. Each of these nodes is the ash-free dry mass, the relative ash-free dry mass of these different functional groups. The arrows represent energy fluxes in joules per meter squared per second. And those are just relative again. So the nodes are relative to each other in size and the arrows are relative to each other in size. And I just want to direct you to a couple interesting results. The first is that when you go from invasion, the top row, to post-invasion, the bottom row, what you'll see is a distinct change in the relative size of the mesopredator node in this teal color to small fishes in yellow. And that changes, the direction is a little different when you look at the seasonal change, but it's a stark decrease compared to pre-invasion. And additionally, perhaps unsurprisingly, what you see is when you go from pre-invasion to post-invasion is an increase in the thickness of these arrows entering the mesopredator node. And this is a little more apparent in this next figure. So this is the energy going out of each of those nodes, colored again by those same functional groups. So yellow is the small fishes. We have the pre-invasion wet season on the left, the post-invasion wet season on the right. And you can see a large decrease in the amount of energy going out of small fishes. So that's saying that fewer higher trophic level species are now consuming those smaller fishes. We see the same pattern in these next two when you compare the pre-dry season to the post-dry season. Again, the amount of yellow gets much smaller in those bars. Now we have the energy influx. So this is the energy going into each node. And again, we have the pre-wet season, post-wet season, and then the dry season, pre and post comparison as well. And here what I want to direct your attention to is in addition to these shifts for the small fishes, post-invasion, we see an increase into the amount of energy going into this mesopredator node. And this mesopredator node includes a variety of species, but also includes these invasive cichlids, such as the African jewelfish. So overall, what did we find? We thought that native species would consume fewer prey that the jewelfish also consume. We did find widespread trophic disruption, but it was a little more complex than native taxa just eating fewer prey that the jewelfish also eat. It was a little more complex than that. When we looked at trophic dispersion, we thought we'd see increased niche sizes. That's what we saw with stomach contents. That's not what we saw with stable isotopes. With both, we saw widespread trophic dispersion. Post-invasion, when we compared spatiotemporal variation to trophic disruption, we found that trophic disruption had a magnitude over 2 times that on average of the spatiotemporal variation. And then when we thought native taxa would rely primarily on detrital energy, what we found that was that post-invasion, almost half of taxa saw an increase in their autotrophic energy use. And then finally, we did see this shift in energy fluxes through the food web after the invasion of the jewelfish, with energy going away from small fishes and towards these mesopredators. So is this a new alternate state in our ecosystem? In chapter 1, we looked at how food webs changed in space and in time, and we found that these drivers piscivory, tidal consumption, and flexible omnivory resulted in shifts in trophic displacement, which was ubiquitous, trophic dispersion, which was common across space and time, and shifts in trophic position were less common and often decoupled from these shifts in diet and in niche in space and in time. In chapter 2, we mixed in ecosystem engineers, and we saw that there was this interaction between changes to the physical habitat and how that interacted with seasonality and environmental stress from seasonality, which is the stress gradient hypothesis. We got a little assist from the niche variation hypothesis to trophic dispersion, these changes in niche size. And we found mixed support for the stress gradient hypothesis, but what we stumbled upon serendipitously were these interesting biotic interactions perhaps, and the relative importance of consumptive versus competitive effects. And then in chapter 3, we added in invasive species, and we found that the African jewelfish invasion has changed food web structure and function, it's altered them. And this leads to the question, is this a new alternate state for this ecosystem? And how will this impact, excuse me, and it seems convincing that this is an alternate state, given the changes in the food web structure and the energy fluxes in the top left, and the changes in community composition, which have been sustained since the decrease in African jewelfish relative abundance several years ago. This community data runs through the end of 2021, several years after the crash of the jewelfish population. So what does all of this mean for the stability and the resilience of this ecosystem? What does it mean for its ability to produce prey, aquatic prey for the iconic wading birds of the Everglades? And after we get a few more years of data, will we see that community composition shift back to that top, bottom left to top right variation that we saw pre-jewelfish invasion? So lots of work left to be done. But with that, I've got a whole bunch of acknowledgements. First, my advisor, Joel, this work all came together because I worked here as a technician for 8 months prior to starting grad school. And we were at the LTER meeting, and I pitched Joel kind of the idea for all this food web work. He goes, wow, that's a great idea. There's a grant due tomorrow at 5 p.m. So I get about a 4 a.m. email from Joel, which is not uncommon. And it's this grant proposal that's ready to go. Tell me what you think. And that is what got all this work going. Of course, Bill Loftus, who I mentioned before, this work would not have been possible without Bill laying the groundwork far before I was born. Bradley Strickland, he's a good friend, great collaborator, great coworker who did a lot of this work together. Numerous research technicians and volunteers. I did not process 4,000 stable isotope samples. I would still be working on them. So that work was invaluable. Of course, all of my committee members. I've got a lot of friends and family and folks from different stages of life that have tuned in today. So very appreciative of all of that. And of course, all of our funding sources. And with that, I'm happy to take any questions.
Let me mention that our standard procedure is that the folks for the committee kind of hold back. And this is the time for public questions. And Peter will be happy to answer those. And then when the public questioning period kind of wanes, then those folks will be able to leave. And then the committee will meet just separately with Peter and discuss his dissertation at that time. So please open the floor for questions. Peter, call on people as you see fit.
Yeah, we'll start with questions in the room. Any questions in the room? Yeah. Sure.
Peter, please repeat the questions because we can't hear them on the video.
Yeah, so we have been asked to go back to the community composition slide. Let me just go out of presenter mode, I guess. So we're going back to our community composition slide here. There's a couple slides, so. Here it is. Yeah. Yeah. So the question is about the community composition through time. And if we were looking to see changes in, as a result of the jewelfish invasion, and then what statistical comparisons did we do, I guess, for the hydrology data? We did, it was just an ANOVA, it was a permuted ANOVA on the hydro period, for each year, and the years were divided into these the same groupings, the same time ranges, and those time ranges were compared. So in Shark River Slough, the mean hydro period, number of days with water greater than or equal to 5 centimeters, was not statistically different across those time periods for those two sites.
Any difference?
Yeah. So the next part of the question is, were you kind of surprised by that? And how does that factor into all this restoration that's been happening? So the answer is that a lot of these sites in Shark River Slough, that are the long-term monitoring sites, were selected because they're in sloughs. They're at a relatively small spatial scale. They're longer hydro period. And I suspect that if you look at things like depth, average depth through the year and things like that, you'll see a lot of change in those variables. But the hydro periods were already relatively long, so there wasn't necessarily that much more for them to increase. You're cut off at 365.
Okay, so these are generally
Yeah, so the question is, are these sites relatively longer hydro period and increased flow won't change their hydro period that much? There's a lot of nuance into this. The hydrologists will not like my answer. The short answer, for the sake of this presentation and for our purposes, no, not really. Any other, John?
Well, because you don't get 21 sites model. You're only looking at spectral site 6 is a longer hydro period than starting to 50 years. I know you don't have data from the sites, so I understand why you take those two, but you brought up at the end this shift, a change in the structure and the thing, but basically all bold statement says it's happening at the sites. What do you think happening at the shorter hydro period sites that basically dry out at the turnover some six?
Well, so the yeah.
So what do you expect?
Right. So the question broadly is, what would I expect out of sites that are more hydrologically variable than the two that were selected because they have community and hydrology data going back to 1978? And well, the answer is that the shorter hydro period sites in other regions of the Everglades didn't have a jewelfish invasion like this. So we were not interested in those for the sake of this. But I did do some of these hydro period analyses that included all of Everglades National Park sites. And when you add in sites from shorter hydro period areas, you do see changes in hydro period.
I'll leave that. The last one. My question is, you started this by saying the static, talking about the idea that fluctuations might be static. It's sort of like an inference we make when you look at those connection webs, and maybe that we can sort of paradigm of static webs. You showed us that the regularity is that COVID positions change, especially dispersions that now overlap in space and time. But I'm actually interested in the few taxa, small fractions that. Is there any regularity to what those organisms are. By size classes or types of ships. Things don't move.
Yeah, so the question is, there was a lot of demonstrated variation in different aspects of trophic ecology, the niche, the diet, trophic position. Is there any commonality among taxa that did not shift? And that is a great question that I actually haven't looked into because I've been so interested in the shifts and what's causing the shifts and who's shifting that I haven't asked the opposite question of who's not shifting.
I just wanted to see
Yeah.
bigger facts there. Yes.
Yeah, I do think off the top of my head, without going back and looking at it, I do think it is larger taxa that tend not to shift as much. But I think it also depends on the comparison, whether it's habitats or whether it's seasons. Anybody else in the room? We see what's happening on Zoom. We've got a couple of questions in the chat, or at least one question in the chat. How does that explanation of no hydroperiod change mess with the results of studies like this one by Joel that demonstrated change at these specific sites, hydrology and fish assemblage composition and abundance resulting from experimental water deliveries? So hydrology is a major driver in the Everglades. Let me just be clear. Looking at hydroperiod is a massive oversimplification of hydrology in the Everglades. We have food web data, pre-invasion, wet and dry season, and we have it post-invasion, wet and dry season. So a lot of like Joel's work has looked at these continuous hydrologic variables like the time since the marsh last dried, and that variable has been shown to have a lot of explanatory power on fish populations and communities in the Everglades. We can do a little bit of that with diet, with our contemporary diets that we collected, and look at how diets change relative to the marsh drying down. But we just can't plot through time at all these different points what the diets are and how these different more complex hydrologic variables correspond to diets because we don't have the diet data that we would need to do that through time at multiple years. But a hydroperiod, I will say, is also an important indicator of the type of marsh in the Everglades and what primary producers are there. And then the rest of the food web flows up from those primary producers. And we know that short hydroperiod and long hydroperiod marshes have fundamentally different food webs because of the differences in the biogeochemistry of those marshes. So while it is not as intricate as some other hydrological variables, it's still important.
If any other factors come to
Any other factors that correspond exactly with the boom and bust of the jewelfish population? Yeah. So the question is, sorry, we had another question in the room and then I'll get this question in the chat. Are there any other possible explanations for the results that we found or any other variables that might have some kind of explanatory power? Well, I'm sure there are lots of variables that have a non-zero explanatory power, but especially for the folks that have worked in this system, it was evident to everybody that when the jewelfish invasion and their population exploded in 2012 following a couple of disturbance events, that something had changed. Have since gone down and showed in a couple of plots how they went back to sort of booms in 2012 were very high for several years, and by the beginning of calendar year 2018 had subsided at most sites back to this pre-boom level of a relatively low density. So that question was, so even at 2018 and 2019 at a relatively low density, it was different than pre-invasion. The answer to that is yes. And then it's worth pointing out that our throw traps, our long-term monitoring sampling gear is bad at catching cichlids across the board. We don't have that quantified and documented or published, but anybody you talk to who has worked with throw traps will be able to tell you that. Cichlids are also good at avoiding electrofishing. So all of our ways to collect these fish probably undersample them. And when we went out to collect fish for this project in 2018 and 2019, in about 2 hours, we caught 50 jewelfish. So it's not like even at these low densities, they were exactly hard for us to catch. So we've got a question in the chat about what caused the jewelfish populations to crash. That is the million-dollar question. This boom-bust dynamic is relatively common for some invasive species. You see it in a variety of systems, and it is a whole area of study unto itself. It could be one thing that has been speculated upon is that the disturbance events in the years preceding the increase in jewelfish population also led to a decrease in native predators. And one of those was a cold weather. There were 2 droughts, with the 2 severe droughts in the 3 to 3.5 years prior to this jewelfish increase. And about 2 years beforehand, there was a cold weather event. We got below freezing. So that below freezing is going to knock back a lot of your non-native fishes. And then those 2 droughts are going to knock back your larger predatory fishes. So the combination of those 2 things is what we think led to the jewelfish increase. And then that crash could be the result of the native predators returning to sort of a pre-disturbance abundance that they were at. But there's a lot of work that still needs to be done on that. We got a question in the room. How big is a jewelfish? A jewelfish, I think my video is still on, is about this big. When we throw trap, we don't keep fish larger than 8 centimeters, and when we electrofish, we don't keep fish smaller than 8 centimeters for the long-term monitoring. So basically, we don't keep jewelfish when we electrofish because they're less than 8 centimeters. That would be a massive jewelfish. Any other questions on Zoom or in the room? All right, well, it looks like we're good on questions. So committee members, hang around. Everybody else, thanks for coming. Thanks for tuning in. I'll go ahead and stop recording now too.